My name is Rashmi Halker Singh. I'm the Deputy Editor of Headache. It's my pleasure to be joined by two of the recipients of this year's Basic Science Award, Dr. Mark Burish and Dr. Sally Yu. They're two of the authors of a recent publication entitled Regulation of Headache Response and Transcriptomic Network by the Trigeminal Ganglion Clock. Welcome to you both. Congratulations on this award as well.
Thank you. We're honored.
First of all, can you tell me about your paper? Tell me what you did and what prompted you to do this work.
So this started just because we've noticed that a few different headache syndromes seem to have a daily rhythm to them. Cluster headache, the most well-known, but there's some features to migraine and even trigeminal neuralgia. So we were wondering if that had kind of a molecular correlate, a behavioral correlate. There's something that was timing the pain. And so I do a lot of cluster headache research. Sally does a lot of circadian biology research, and so we kind of combine forces and also work with a few other people, Amina Pradhan, who does a lot of pain research, headache research, and a couple other people who do more kind of transcriptomics. And so we put this together to try to figure out if the trigeminal ganglion had a circadian rhythm to it. And we found that it did.
So I think that's really fascinating. It goes along with what we see in clinical practice as well. Our patients are commonly telling us about the timing of their attacks from cluster and also with migraine. So share with me what you found from your work. What were the results of your research?
So I'm a circadian biologist and I study circadian rhythm entire my career. And circadian rhythm is basically your endogenous clock, built-in clock that you have. Tell your body to what to do in what time point to maximize your efficiency of physiology because we are all living in Earth, 24-hour cycle of day and night. So our system evolved to adjust our physiology to it. So when we eat, we are ready to digest. When we sleep, our muscle and every body don't produce energy because we don't need it. So this system is innate. And our curiosity was how this innate clock system is regulating the pain response because various type of pain has circadian rhythm. And me and Mark work was really attacking how circadian rhythm is regulating headache pain. So we decide to look for the headache center trigeminal ganglion, and looking trigeminal ganglion has innate circadian clock. And if it has circadian clock, how it regulates the pain response. In terms of mouse genetics, molecular, and pharmacogenetics, we really dig the mechanism of how circadian clock is regulating pain response. And also in terms of translational direction, we wanted to see that how all this knowledge can actually help the patient that when is the best time for taking this specific drug because this drug target expresses a certain time of the day peak time.
So all of that is very fascinating. And before you share with me, clinical correlations about these things, I think one thing I want to add is this also gives a lot of validity to what our patients are telling us, right? Because our patients share that they have this time correlation with their attacks, whether it be migraine or whether it be cluster, and now we have some biological basis for that. And I think that really helps to strengthen what their clinical experience is. So that's really, really fascinating. Um, you shared in your paper and what you just shared right now about, maybe we can take some of these findings and apply this to clinical practice and maybe give our patients some recommendations about timing of medications or other things. Can you share some of that with us right now too?
Sure. So this is in internal medicine, they do this all the time. They give blood pressure medications at certain times a day because blood pressure kind of spikes at different times. So we're trying to bring this over more from a headache side of things. So like Sally was talking about, there's a series of genes that kind of turn each other on and off over 24 hours, but they then signal other genes to turn up and down over 24 hours. So they kind of are the central clock or the main clock, and then they turn other genes up and down. So we looked at all the genes that are cycling either in a normal mouse or in the headache mouse. This is from Amina Pradhan's nitroglycerin model. And as you know, if you inject nitroglycerin into a migraine patient, they get migraine, cluster headache patient, they get cluster headache. It's a very good human model. But the idea here is if there's a gene there that's cycling, is there a drug that targets the gene? And so we took basically kind of I guess the Venn diagram here between all of the circadian kind of cycling genes, and then all of the medications that are guideline approved for cluster headache, migraine, or trigeminal neuralgia. And we found there were about 10 drugs that do target genes that kind of cycle over 24 hours. So this is kind of an initial idea of maybe could we start to study the best time to take these medicines.
I think that's really fascinating work, and sets us up for, additional research in this area as well. Um, do you have any additional thoughts you want to share with our viewers?
Oh, I'm very excited to start this. We have been working together for many years, but we really feel like we now have really great grip to crack more mechanism of circadian regulation of headache pain. And we're very excited to do more to understand that how we can actually apply this basic knowledge to the patient treatment.
Yeah, I think, like Sally said, this has kind of given us a very clear direction in what we do next from an animal side. We've kind of started to create an animal model of circadian headache. And so we're excited for what comes next.
That's really fascinating. Thank you so much for all the research you're doing. This was really advancing the science of headache medicine, and we really, really appreciate you both and your colleagues.
Thank you very much for having us on. And again, we're honored to receive this.
Thank you.